DNA double-strand breaks, DSBs, the most severe type of DNA damage, leads to apoptosis, gene mutation, and carcinogenesis if left unattended. To overcome defective DNA DSBs repair, a strong fundamental understanding about the relevant underlying mechanisms must be acquired. In DSBs repair, the mRNA complex acts as a sensor towards DSBs and also an initiator of DNA damage responses. However, when DSBs is formed, it is still elusive with regards to how the mRNA complex senses and binds to DNA rapidly. In an international collaboration with Sun Yat-sen University Cancer Center in China, this study discloses the underlying mechanism of DSB repair carried out by mRNA complex interacting protein, MRNIP. In intrinsically disordered region, the MRNIP forms condensates which then concentrates mRNA complex into liquid-like droplets. When DSBs are formed and sensed, the mRNIP condensates proceed swiftly to the damaged DNA and promote the binding of damaged DNA to the concentrated mRNA complex. This results in the autophosphorylation of ataxia telangiectasia mutated followed by an accelerated DNA damage response and DSB end resection, promoting the homologous recombination-mediated DSB repair. The output of this study is an essential reference for future research which target to overcome diseases related to defective DSBs repairs. By providing significant insights on the pathway of DSBs repair, this study is very well aligned with UN SDG 3, good health and well-being.